course, we all understand the context of climate change and this challenge of carbon abatement. And if we reflect on, for instance, the Stern Review, the trajectory for stabilisation at 450 parts per million of atmospheric carbon dioxide is a significant challenge. So we're talking big numbers, 135 billion tonnes of CO2 abatement products that need to be coming online between now and 2050. Garno reflected on this in the local context, of course, and his proposition last year in the update report was that we needed to reduce 25% on 2000 levels by 2020 to target 450 parts per million, or our contribution to, and that's 272 megatons of CO2 equivalent, so big numbers. The concurrent thing that runs alongside this, of course, is that it's not a static picture, of course. The IEA expects that energy consumption and demand is going to grow by 40% over the next 2 decades, so it's a pretty frightening statistic. Now, I'm from Melbourne, so when we talk about scale, we talk about the MCG. Here in Sydney, you talk about Sydney Harbours. I don't know how many that would take to fill, but it's a hell of a big number. The strategic opportunity that exists, I see, is for biomass, and there's a contribution that I see biomass as organic matter that can actually contribute to that carbon abatement challenge. Plants, of course, take CO2 out of the atmosphere as they photosynthesise, and this biomass can be harvested, processed and refined into a whole number of petrochemical substitutes and valuable products of all different types, and I'll touch on some of those later in the presentation. But the key point I wanted to make here is that biomass is our only option for substitution of material feedstocks. So you can have all the wind turbines and all the solar panels that you want to generate electricity, but you'll never have something that gives you an actual physical material that you can substitute without biomass. So biomass has that unique position to occupy in the sustainable future in providing a material feedstock substitute. But of course, there's many challenges associated with delivering on that sort of volume. Don't worry too much about the text here, and I'll go through with this little jigger here to sort of point out the features of this diagram. But this was an analysis that I did that looked at global food production, population projections, and trying to understand the tension that exists with looking at first generation oilseed crops and food crops to be actually diverted towards bioenergy production. So there's this big food versus fuel debate that you probably would have all heard about. So what we've got here is 3 different population projections. This is based on a UN population projection, so that's the sort of low end of the scale, the medium and the high. And what we have here is 3 different scenarios which are based on a vegetarian diet, so for argument's sake that the world is purely vegetarian, a moderate protein diet and a high protein diet. And the other feature in this diagram is what we call a low external input production threshold, where we're using organic farming methods, we're not using any kind of, limited amounts of mechanisation or artificial fertiliser applications. So very much where a lot of the developed world, that sort of level of technology versus a very highly, genetic modification, etc., high application of fertiliser, high intensive irrigation, etc. for production. Cut a long story short, what it tells us is that even on a moderate protein diet, where the average global citizen is actually consuming, a proportion of meat in their diet, we're getting to a point where we're actually reaching, a problem there between the actual capacity of the production system at the low external input threshold anyway, and the current global population. So what that means is, to cut a long story short from this diagram, is that if you do that analysis, you understand that there really isn't any available or significant amounts of available agricultural land that would be otherwise diverted to food production. If we're going to feed the human population, we're going to have a real problem if we're diverting corn and these sorts of crops to the production of biofuels and bioenergy type products. So what are microalgae and why am I interested in it? There's some little happy snaps from my own research over the last few years. But microalgae, of course, are single-celled aquatic plants, and they're very, very small and range generally in size between 5 to 100 micron. One of the advantages of actually considering microalgae as a source of biomass is that they don't compete with productive agricultural land, of course, because they're growing in water bodies or existing food and feed commodity products and markets. They're incredibly productive as a source of biomass per unit area, so that's one of the things that has always attracted interest to this particular area. And they're highly efficient in terms of their growth, so they can double their mass roughly every 1 to 2 days, although they do tend to grow in fairly low densities in very large volumes of suspension. So it's the proverbial sort of needle in the haystack in an aqueous form. But the rough rule of thumb is that they take about 1.83 tonnes of carbon dioxide, or require that, to produce 1 tonne of biomass. So if you do the maths on that, you can multiply it out, and it's a very easy kind of rule of thumb that you can use to get a bit of a sense of the scale and the opportunity that might pertain to. Once you've harvested that biomass, there's a whole lot of different things that you can do with it, and I'm just touching on a couple of things today. But certainly there's all sorts of processing options and products and things that you can do with biomass. Significantly, you can actually utilise wastewater streams and also waste gaseous emissions. So flue gas capture is a really great opportunity for coupling with microalgae, and the company I'm working for is doing exactly that at Tarong Power Station in southeast Queensland at the moment. But it presents an opportunity to create a very high value source of sustainable biomass. Conveniently, they greatly increase their rate of reproduction when they're given supplementary CO2. So they'll happily grow and photosynthesise in water bodies as they are. But of course, if you artificially supplement them with additional CO2, then that'll just kick off a new phase of growth to a saturation point, of course. And the commercial viability of that whole proposition on a large industrial scale actually also depends on that productivity boost. So it's kind of a happy arrangement, I suppose, on both sides. But it's a great opportunity, I see, for industrial ecology, this concept of coupling a co-location of an emissions-intensive industry, such as power generation or stationary industry or heavy industry, and actually, coupling it with a biomass production system to achieve a positive outcome. A lot of you may have heard of the direct extraction of lipids from algae, and there's been a lot of press about that in the last, well, it goes back actually to the 1970s in the oil shocks when the US Department of Energy got pretty interested in this. I don't see that that's a particularly fantastic possibility in future, and I'm happy to talk about that during question time or with any of you that would choose to do so. But that pathway is particularly technically and commercially problematic as I see it, and there's a trade-off in the growth and reproduction of microalgae. So if you think about it, if you're a photosynthesising plant and you're sitting there and you're capturing light and you're taking that energy in and you're channeling that into storage, so lipid production, so fat essentially, or form of, if you like, a storage mechanism, then that by definition means that you're not using that energy to reproduce and grow the culture. So there's a trade-off. So a lot of the traditional work that's been done in this area focused on, oops, this is stuck on, I think. A lot of the work in this area focused on targeting microalgae species that were very, very high in natural lipids to extract for biodiesel production, etc. But by definition, that means they're not growing very quickly. So my interest is in the stuff that you read about when people are cursing on their summer holidays and they're jumping in the water hole or in the Gippsland Lakes, because I'm from Victoria and there's all sorts of blue-green algae gumming up the waterways. I want the stuff that grows like stink, because that means that's the maximal turnover of carbon dioxide and reproduction of that particular source of biomass. So what does it look like as a process flow when you get out into industry? So if we look at sort of aquaculture and farming, there's a prawn farm that I'm working on up in far north Queensland at the moment, so remediating the water, taking out the nitrogen and phosphorus, growing algae in that scenario. You have a mining or industrial type operation, so a smelter, and of course stationary power production or energy companies. Essentially the model is fairly similar. So you're taking effluent water, carbon dioxide and sunlight, of course, in what we call a phototrophic system, and you're allowing algae to do what it does best and has done for billions of years. It photosynthesises and creates biomass, and of course the byproduct of photosynthesis, as we all know, is oxygen, and part of that process is it cleans up the water at the same time, so you can recycle that water, and for a lot of these guys it enables them to discharge safely at a safe level to, for instance, in the case of this prawn farm, it's on the Great Barrier Reef Marine Park, so it enables them to safely discharge without incurring a problem. And then the downstream impact of that, of course, is all of these different potential products that you can derive from that biomass. The one that I'm really going to focus on today that is significant in the context of my research is this biochar pathway, and there's only one route to get into that. But before I go any further, I wanted to share with you, this is a vision. This is not my idea, it's not a new idea. The father of photobiochemistry, Giacomo Chimichine, I hope there's no Italians in the audience that I'm offending with my poor Italian pronunciation, but it's a fantastic, inspiring story for me. So he wrote back in 1912 in Science, "On the arid lands there will spring up industrial colonies without smoke and without smokestacks. Forests of glass tubes will extend over the plains and glass buildings will rise everywhere. Inside of these will take place the photochemical processes that have hitherto been the guarded secret of the plants, but that will have been mastered by human industry, which will know how to make them bear even more abundant fruit than nature, for nature is not in a hurry and mankind is." It's a fantastic kind of vision, and funnily enough, exactly 100 years ago. So what is this technique that enables you to get to that biochar product? It's a technique called pyrolysis. It's one of the many pathways of conversion of that biomass product to a range of different valuable byproducts. But pyrolysis is a process essentially in which a material, usually biomass, but it's also used in the waste treatment industry, plastics, etc. But you can essentially degrade a material in a furnace by heating it without oxygen to avoid combusting it. And what happens essentially as you heat it up systematically is you get a decomposition into three primary sort of products or fractions. So you get a component that will be a gas, you get an oil or a liquid, which is essentially like a bio-crude or a bio-oil, and you end up with essentially a residue which we call biochar. And there's two different, very crudely, there's two different mechanisms or process parameters that you can run within a pyrolysis process. One is called fast or flash pyrolysis, which has very, very short residence times of less than a second and heats the material up to over 650 degrees, typically around 700, something like that. And that tends to drive the process towards a maximum production of bio-oil of those three component products. You'll still get the other two, but it's maximum for bio-oil. Whereas what I call slow, or what's called slow pyrolysis, and what I've focused on in my particular research, and it's also referred to as carbonisation, is this process that takes 1 hour plus, very slow heating rates, perhaps 10 degrees per minute, which is essentially what I've run throughout my work, to fairly moderate temperatures, a maximum of 550 degrees, and that produces a maximum amount of biochar. Some of the results, and I'll come back to why that's significant in just a moment. Some of the results with this particular species, Tetraselmis chewi, that I've done my research around. Tetraselmis chewi is a common mariculture species of micro that's used, for instance, to feed oysters, and New South Wales DPI at Port Stephens feed them there to the baby fish and the oyster spat. So it's quite commonly used for that purpose. Some of the analytical results that came out of the work that I've done is that if you look at the actual raw product, they've got a fixed carbon content that's fairly similar to wood, sunflowers and rapeseed, so almost 13% of fixed carbon content. Because this is a marine species of microalgae, so it grows in salt water, has a very high ash content, so somewhere between wheat straw and rice straw. The biogas that evolves through that slow pyrolysis process, fortunately, is slightly more than the energy that's required to actually achieve the thermal decomposition of that material in the first place. So in theory, you can capture that biogas and offset the energy requirements. So it's not quite a self-sustaining process would be the wrong way to describe it, but it doesn't, you can actually utilise the energy value of that biogas to offset the process requirements. In terms of the bio-oil and the liquid, like all pyrolysis oils, it's a pretty dirty, ugly, sticky, potentially acidic looking syrup, and it's usually highly oxygenated, so it's likely to require some upgrade to reduce that oxygen content, but it has a calorific value of about 28 megajoules per kilogram, which is relatively high, particularly for a pyrolytic oil. The biochar, which is converted to 37%, so roughly just over a third of the starting weight of that raw biomass, concentrates the ash, the minerals, from 15% to about 30%, and that's very similar to the work that's been done around some of the seaweeds, the larger macroalgae, and this is actually a colleague at James Cook University that I'm working with now. But the biochar is extremely sodic and very, very saline, but very similar in terms of its behaviour perhaps to a conventional fertiliser in very well-drained soil, because they come in a salt form as well. And as long as you've got well-drained soil, it will essentially wash through the soil over time. The total amount of carbon that is concentrated then in that biochar is about 40%, and that's relates to an organic carbon content of 16%, and that difference of 24 amounts to 9% of weight of the carbon from the original raw algae that can be actually fixed into the soil. So the whole long-term proposition of biochar is about adding it to soil as a bio-carbon capture and sequestration option to countervail the option of geological carbon capture and storage, which I'm sure you've all heard about. Incredibly expensive, incredibly energy intensive. I don't think will ever happen, but we'll wait and see. But certainly provides an alternative biological route to actually synthesise a highly stable form of carbon that will have a residence time in soil that's measured potentially in the thousands, at least hundreds of years, as opposed to the technological fix, which is essentially trying to stuff the genie back into the bottle, that will cost an enormous amount of money. If you look at some of the agronomic properties of this biochar, I won't go through this in detail, but essentially it has some very good sort of fertiliser, offset type or agronomic properties that are valuable in an agronomic context. It's got a very high cation exchange capacity, so it's able to actually, essentially the mobility of the nutrients within that are able to be exchanged very easily. Although I mentioned it was extremely sodic, it does have a very high pH, so it can be used as sort of a similar lime, similar to a lime application in soil. So it has some positive opportunities there for utilisation in an agronomic context, and that's what a lot of the biochar research is actually starting to reveal. So this is using just one form of biomass, being microalgae, to produce something that can actually have this multiplying added value effect by not only sequestering carbon in large volumes in soil, but also adding value to the soil at the same time. This is a photograph, a scanning electron microscope image that was taken courtesy of colleague Tony Morrison up the back there. So thank you, Tony, once again. But one of the interesting things about the surface area of a biochar is it's incredibly, again, we were talking about surface area before, in Mark's presentation, but you get a surface area of roughly 19 metres squared per gram of biochar that's produced. So it's a huge amount of surface area in very small amount of material. And why is that useful or interesting? It's because this is what gives it some of that positive value-adding opportunity in soil, because it creates a happy little home for soil microbes and mycorrhizae and all of these fungi that add to life in soil. So it creates essentially these microhabitats that encourage microbial growth and also actually, funnily enough, rapidly increase the moisture retention capacity of the soil. So it might be a good idea in rural Australia where we've got problems with irrigation and water resources as well. So some potentially really interesting value-adding opportunities. So the basic premise of the research is what I call the carbon pump. It's about combining a very high volume, scalable microalgae biomass cultivation system, which happens to have other adding value opportunities, with a high volume distributed slow pyrolysis processing system that produces biogas, bio-oil, and particularly the biochar for soil remediation and carbon abatement. So the issue really ultimately is, if you combine these two things, could we do it? Yeah, I reckon we probably could. But the biggest concern ultimately, and where I've driven my research to, was yes, we might be able to deal with all of these issues that we've already touched on. Climate change, I didn't mention peak oil, but that would be a familiar theme to many of you. We talked about food versus fuel, nutrient cycling, etc. The issue really is, well, what's the life cycle impact of doing this? If you scale it up, big style, and we're talking gigatonnages of production, tens of thousands of hectares, big farm, broad acre farms, what's the impact of doing it? It's fine in the lab, but what does it look like when you create a whole new industry that's trying to do this? So the question becomes not could we, but should we? So what I have here is kind of a life cycle model, which starts with the microalgae biomass that's being cultivated. And there's a series of steps to get that needle out of the haystack, essentially, and isolate it, because to pyrolyse a material, it needs to be dry. So you need to get it through these dewatering stages and dry it. And then there's a number of different processing routes. So I looked at two different things. One was pyrolysing the raw algae as it is, and the other one was taking out the lipids before you then pyrolyse the residue. And the reason why you might do that is because you can get very high commercial returns for the good lipids. It wouldn't be for transesterifying it into biodiesel, most likely, but I did model that to see what it would look like. So you end up with these products I talked about before, bio-oil, biogas and biochar, or if you directly extract the lipid, then you could transesterify into biodiesel. And I used what was an adapted life cycle assessment method, and this is something that was used or has been used to benchmark a whole range of fast-moving consumer goods products that you can buy at Walmart. There's an organisation in the United States that started to benchmark a whole lot of sort of common consumer products. And this looks at environmental impact categories across a range of indicators. So it's not just about global warming capacity and greenhouse gases, it's about a whole range of other things. So water depletion, eutrophication, acidification, ecotoxicity, ozone depletion, etc. And this is a common approach in life cycle assessment methodology. You take and you rank and assess the impact of a particular process or product or material on all of these chosen environmental impact categories. Then you normalise the data, you weight the data to come up with a single aggregated, what they call an eco-point score. And the reason you do that is that the units of reporting for all of these are quite different and specific to what you're trying to measure. So you need to have some way of putting it all into one single aggregated figure that you can use for a point of comparison. And an eco-point essentially is benchmarked against the average, think of it as a budget. The average Australian citizen has 100 eco-points to spend in a year. So the number of eco-points will relate to the average impact, environmental impact of one Australian citizen in 1 year. So what you end up with is a really positive outcome, a negative outcome in eco-points through global warming, of course, because you're coupling to a power station, you're actually taking CO2 out of the atmosphere that it would otherwise go up into the atmosphere and contribute to the problem. So there's a really good story to be told around global warming and what can be done with this kind of coupling system in a point source emissions context. In terms of other sort of features though, a relative contribution to eco-points has a huge impact of acidification and particularly, and also photochemical smog. And this essentially relates to the utilisation of fertiliser. Because if you think about doing this on an industrial scale, you are going to have to get nutrients from somewhere that's going to drive the growth of the plant. So if you do it on that scale that we're talking about in the gigatonnages, then there's some real questions to be asked. And the production of fertiliser, at least in the conventional sense, is incredibly energy intensive and creates all sorts of ancillary problems. You can see here that there's also, this is a breakdown of the different environmental impact categories and those parts of the life cycle, so the cultivation, primary harvesting, secondary harvesting, tertiary and spray drying. And you can see spray drying in particular is incredibly energy intensive. So this model was built on best available technology, and that's showing a pretty ugly story at the moment. I won't go through any of these as a feature, but what I have done is put into equivalents and eco-points per gigajoule of microalgae to compare it with soybean and canola seeds, so conventional bioenergy crops. And you can see that it's actually somewhere in the middle between those two in terms of the aggregated environmental impact. So you can see there's a really good story again to be told there with microalgae where it's coupled to a power station. But overall there's some other impacts that are far higher. So it comes out at a roughly equivalent level at the moment to that. So what does this mean in a number of different scenarios? When I run it, if you pyrolyse for purely electricity versus electricity and biochar, taking out the oil, pyrolysing the residues, a number of different scenarios that I ran, but you can sort of see some equivalences here with the Queensland electricity grid, for instance. The amount of CO2 equivalent per megawatt hour produced, if you're looking at electricity, is 0.94 tonnes of CO2. Well, if you look at these different scenarios here, you're getting significantly less than that, which doesn't necessarily mean it's a sustainable option, by the way. The biochar I benchmarked against existing NPKs fertiliser, so standard fertiliser, and how much tonnes of CO2 equivalent is produced to produce a tonne of that product. So you can see that actually in this scenario it's getting pretty close in equivalence to the same amount of impact of producing 1 tonne of conventional fertiliser. And then if you're looking at biodiesel as an end product, again, soy biodiesel comes out a lot better actually in this case than the oil extraction from microalgae. So some of the conclusions that I've come to in this work, and this is, I hope, well, it's been submitted for publication and I hope comes back with good news fairly soon, but you get some, as I mentioned before, a good credit for the photosynthesis and the biochar production, because that is net drawing down a certain amount of CO2, but unfortunately, when you compare it with the overall impact of the total system and the value chain that needs to be put in place at scale to actually produce these outcomes, it really pales in comparison to the impact of that broader system. But the good news is it comes out roughly comparable with common bioenergy products that we deal with every day at the moment, that have a lot of those associated problems of food versus fuel. And I think ultimately that's based on, as I said, best available technology and known adapted technology that's used today, but I think there's huge opportunities for technical process innovation that will improve the life cycle impact of micro in all of those scenarios. Particularly you saw with the process heat, or sorry, I beg your pardon, with the spray drying. If you think about a power station, well, power stations are much better at producing the heat than they are at producing electricity. About 2/3 of the energy content of the material that goes in mostly goes up into the atmosphere as heat. So there's lots of standby heat opportunities that you could utilise, provided you can engineer that and happily do that with the power station's blessing, that would significantly, for instance, reduce a lot of the embodied energy that's being used in that process. But finally, I just wanted to underscore that this process is the only means to achieve a value-adding biological carbon capture and sequestration solution. So it's something that needs to be innovated, I believe, and can be innovated, but there's a lot of work to do, I think, is probably the outcome of the initial study based on known technology and scalability of systems as we know them today. So thank you for letting me share that with you, and if there's any questions.